MBBS, and Dr. Deepu Shivadas from KFRI to kindly occupy your seats on the dais. To welcome the gathering, may I request Dr. K. Vijay Kumar, Principal Scientist, KSCST.
Respected Dattan Sir, Directors of R&D Centers, dear students, delegates, and my dear KSYSA Awardees. Kerala State Council for Science, Technology and Environment has instituted this award, Kerala State Young Scientist Award, from 2012 onwards. So we awarded this prestigious award to 60 young scientists till this 2024 for their outstanding contributions in science and technology. They can nominations can be submitted to KCST under 14 subject areas for this award. And there will be a screening and then a selection committee for this award. And the awardees will get a cash prize of 50,000. And they are also eligible for submitting a project proposal. And after peer reviewing, they will be awarded a maximum grant of 50 lakhs. And also, they are eligible for a travel grant for attending an international conference. So this award was instituted by KCST for attracting young talents in Kerala for performing, contributing to the development of Kerala. So in this category, today we are having two young scientists with us, Dr. Vrinda Mukundan and Dr. Harish. Dr. Vrinda Mukundan is working at the National Center for Earth Science Studies, Thiruvananthapuram, and Dr. Harish V.S. is working at Malabar Botanic Garden and Institute for Plant Sciences. So this is a grand occasion for us to welcome these two young scientists to this 37th Kerala Science Congress. And also, I am proud to introduce these two young scientists to you for their award presentation here today. And I also offering our formal welcome to Dr. Deepu Shivadas and also all the audience here and the dignitaries and all the students for attending this program. Thank you.
Thank you, sir. We'll now move on to the presentation by the KSYSA awardees. To begin, we start with Dr. Vrinda Mukundan, DST Inspire faculty from the National Center for Earth Science Studies, Thiruvananthapuram. Over to Dr. Vrinda for the presentation.
A warm good evening to all of you. I am Dr. Vrinda Mukundan, and I have been a researcher in the field of space and planetary sciences for the past 12 years. So my research basically focuses on understanding a particular region of the planetary atmospheres called upper atmospheres. So you might have noticed I said upper atmospheres specifically, not atmospheres. So let's understand what this upper atmospheres and what makes it different from the lower atmospheric region. So let's take a look at the Earth's atmosphere. The Earth's atmosphere is divided into different layers like troposphere, stratosphere, mesosphere, etc., depending upon how the air temperature in this region behaves. Now, those regions above an altitude of around 90 kilometers is usually referred to as the upper atmosphere. This region is characterized by high air temperatures. And this occurs because of the very low density of the atmosphere that occurs at this altitude. Because of the low atmospheric density, even a small amount of solar radiation can raise the temperature to very high values. So that's why you get to see high temperature in the upper atmospheric region. So in addition to this high temperature, another characteristic feature of the upper atmosphere is the existence of a charged region called ionosphere. So by charged I means generally in the atmosphere we talk about neutral gases, neutral atoms and molecules. Whereas in the ionosphere, what happens is when the solar ultraviolet radiations, which are basically high energy radiation, interact with these neutral gas particles, it ionizes them. This process is called photoionization. So during this photoionization process, the neutral gas particles get converted to positive ions and free electrons, thus leading to the formation of ionosphere. Thus in the ionosphere, along with the neutral particles, a significant fraction of the particles are in a charged state. So now this atmosphere or upper atmosphere and neutral atmosphere, or be it ionosphere, all these terms are not something which is confined to Earth's atmosphere. So if you look at our solar system, there are several planets which has got a significant atmosphere. All these atmospheres will be having an ionosphere as well. So the past 7 years, I have been working on the ionosphere of our nearest neighbor, which is Mars. So over the next 10 minutes, I will take you through my research journey through the Martian ionosphere, and will discuss with you some of the key research findings that I have made. So speaking about Mars, Mars has got a very thin atmosphere. The atmosphere is primarily composed of carbon dioxide, which constitutes about 90%, more than 90% of the total atmospheric composition. Along with CO2, there exist other minor gases like atomic oxygen, argon, nitrogen, etc. When solar ultraviolet radiation interact with these neutral gas particles in the upper atmosphere, as I said before, what happens? Photoionization. So during this photoionization, these neutral particles get converted to positive ions and free electrons, leading to the formation of an ionosphere. So what I have shown over here is something called the density profiles of the free ions, positive ions and free electrons on the Martian dayside ionosphere. So density profiles is nothing but it simply tells you how the densities of these charged particles are changing with respect to altitude. That is, as you go to higher and higher altitudes, how many of these charged particles you will be able to see at each altitude. So that is what basically density profiles tell you. So as you can see, the most dominant ion in the Martian dayside ionosphere is O2+, followed by minor ions like CO2+, O+, and N+. That thick black curve that I have shown over here is something called the free electron density profiles, as I said before, which tells you how the density of the free electrons is varying as you go to higher and higher altitude. So this electron density profile is something like a characteristic signature of a planet's ionosphere. Simply by looking at the electron density profile itself, you will get to understand many properties of a planetary ionosphere. So if you take a close look, you would be able to see two peaks in the Martian ionosphere. The major peak is referred to as the M2 peak, and it occurs at an altitude of 125 kilometers. So it is at this M2 region you get to see maximum electron density in the ionosphere of Mars. So below the M2 peak, there is a minor peak that is called the M1 peak, and it occurs at around 110 kilometers. So this is the typical structure of the Martian dayside ionosphere. But the reality is, you don't get to observe the same set of features whenever you observe the Martian ionosphere. Sometimes the M2 and the M1 peaks can have higher densities, sometimes it can have lower densities. Sometimes these peaks shifts to higher altitudes, or sometimes it shifts to lower altitudes. And occasionally you will get to see transient layers that appears above the M2 peak. So all this indicates that Martian ionosphere is highly complex and dynamic, with plasma densities showing a very high degree of variability. And this variability basically occurs due to the changes in the chemistry, energetics, and dynamics of the Martian ionosphere. Or in very simple terms, I can say that there are basically two reasons for this dynamic nature. One, the changes are happening in the neutral atmosphere. Two, the changes are happening in the amount of UV radiation that the planet is receiving. That is basically, it is the combination of this neutral atmosphere and the UV radiation which is leading to the formation of this ionosphere, isn't it? So any change happens in these two factors, you will definitely get to see a signature on the ionosphere. Now, why these two parameters should change? So there are several situations during which these two parameters, that is the neutral atmosphere and the UV radiation, can change. For example, when there occurs seasonal changes, the Martian neutral atmosphere get affected. Or there occurs dust storms. You might have already heard about dust storms occurring on Earth, isn't it? So dust storms are not something which is confined to our planet. It happens actually plenty of them happens in Mars as well. So when dust storms occur in Mars, the neutral atmosphere will get affected. So this signature will be seen on the ionosphere. Similarly, coming to UV radiation. So when UV radiation is something which can change drastically over a solar cycle. So solar cycle means it's a natural cycle of the sun which occurs for a duration of 11 years. So it happens in every 11 years. So during this duration, there can occur significant change in the amount of radiation, especially UV and X-ray radiation emitted by the sun. So when UV radiation can change during a solar cycle, its signature will also be seen on the ionosphere. So all these factors makes the ionosphere very dynamic in nature. Now we come to the question why we should be actually bothered about this Martian ionospheric variability. Martian ionosphere is variable, okay, then let it be so. Why we should be worried about it? Actually, the basic intention of understanding the Martian ionosphere is to understand, or Martian ionosphere is to understand how the atmosphere of Mars evolved. The researchers have already shown that in the long past period, Mars used to have a very thick atmosphere where the temperature was significantly high and the atmosphere was much more wetter. But now Mars is having a very cold, thin and arid atmosphere. So how the atmosphere deteriorated into its present form? So there are several factors like the low gravity of the planet, also the absence of a global magnetic field which resulted in its deterioration. When a planet lacks a global magnetic field, we know Earth has got a magnetic field, isn't it? But Mars doesn't have that. So when a planet doesn't have a global magnetic field, what happens is the atmosphere of the planet will be continuously exposed to something called the solar wind, which is a strong stream of energetic charged particles from the sun. So when this energetic charged particles interact with the planet atmosphere, it accelerates this atmospheric deterioration process. Now where is ionosphere coming into this picture? This ionosphere acts as an integral boundary between the planet's atmosphere and the solar wind, and it largely controls this atmospheric interaction with the solar wind. So if you want to understand how the atmosphere of Mars deteriorated, you need to understand the ionosphere and its variability. So this need to understand and quantify the variability of the Martian ionosphere on spatial and temporal scales is the main objective behind my research work. For this purpose, I developed a photochemical model. And this model I am using as a tool to extensively analyze and interpret the observations from a particular spacecraft called MAVEN. The MAVEN was a mission sent to Mars by NASA in the year 2015. It was like a contemporary mission with India's Mangalyaan. So this MAVEN mission was a dedicated mission for studying the Martian upper atmosphere and ionosphere. So by combining my photochemical model with MAVEN's observation, I am able to better interpret and deepen our understanding of the Martian ionospheric features. So this figure shows the model that I have developed for studying the Martian desert ionosphere. I am not getting into the intricacies of the model. So all you just need to understand here is the model needs some inputs to work, isn't it? Any model needs some input to work. So for a realistic simulation, what I did is I used the actual MAVEN in situ measured atmospheric parameters of the Martian atmosphere as input to the model and made the model run. And I started getting output and I started analyzing it. And let's see some of the results which I come up with. So once the model was developed, my first intention was to compare the model calculated or model predicted densities or the model predicted results with MAVEN observed atmospheric parameters. So such a model observation comparison is very significant to understand whether you are on the right track. That is, if the model is agreeing with the observation, that means, okay, our understanding is correct. But if there are differences, it again points to the fact that there are lacunae in our understanding and we need to do further research. So when I made such model observation comparisons, I could find two important things. The first is my model struggled to reproduce the observed densities of the most dominant ions, O2 plus and CO2 plus, in the Martian desert ionosphere when I used MAVEN measured in situ parameters as the model input. So, and I figured out that this occurs because of the electron temperature with the MAVEN spacecraft is measuring that I am using as input to the model. So my study actually pointed to the fact that there is something missing with the MAVEN's measurement of electron temperature and it need to be re-scrutinized. Similar is the case with another parameter called ionization efficiency, which I have shown over here. So this ionization efficiency basically tells you how effectively the electrons generated in the Martian ionosphere can cause further ionization. So in this case also, I found out that when I calculate this ionization efficiency value using MAVEN measured parameter called photoelectron flux, I get a straight curve, a straight constant value which is actually contrary to the theory. As per theory, this particular parameter should increase as you go towards the lower altitude due to various reasons. Again, I figured out that it is due to the MAVEN's limitation in the energy resolution of measuring this parameter called photoelectron flux. So to address this problem, I came up with an enhanced ionization efficiency profile, which is a very useful parameters for modelers who are concentrating on this Martian ionospheric research. So these two work together highlights the power of a model to understand the lacunas in the observations made by a spacecraft, and is actually pointing to the fact that modeling and spacecraft observations should go hand in hand to have a better understanding of the planetary atmospheres. Now, the next step, in the next step, I use the model to bridge the observational gaps. Means like a spacecraft always has spatial and temporal limitations. It can make observations only at one particular point at one particular time. So if you want to understand what was happening at some other point at the same time, you need to simulate it. So one such situation happened during the 2018 global dust storm event. So during this particular dust storm event, the MAVEN spacecraft was observing the northern hemisphere of the planet. So at that time, we wanted to know what was happening at the southern hemisphere. So since we didn't get observations from the spacecraft, I simulated the ionosphere of the southern hemisphere during this dust storm event. And I found something interesting. For the ionosphere in the southern hemisphere to respond to this dust storm, it took some time. There was a time delay. That is, it took around 3 weeks for the southern hemisphere ionosphere to respond to this event. So this actually points to the fact that when an event like dust storm is taking place, a global dust storm is taking place in the planet Mars, the atmosphere of the planet is not responding altogether. There are variations in the behavior of the atmosphere of the planet at different regions, depending upon whether you are looking at the northern hemisphere or southern hemisphere, like that. So this kind of findings has got important implications when you plan a mission for planets like Mars. So this particular work highlights how the model was used to support the spacecraft observations. So in the next step, I started using the model for interpreting the observations from the MAVEN spacecraft. So this particular work also concentrated on a on a dust storm, but this time it was a regional dust storm that occurred in the year 2016. So during this particular dust storm event, it was observed that the ionospheric peak, which we discussed earlier, enhanced unusually by around 20 kilometers, which is twice the expected value during such events. So using my model, I investigated the reason for this dust storm. So usually the researchers were claiming that it was due to that dust storm which caused this unusual enhancement. But with my study, I could prove that it was not due to the dust storm. There occurred another thing called perihelion effect. That is, during the occurrence of this particular dust storm, the Mars was very close to the sun. So very close to sun means it was receiving enhanced solar radiation. That means the atmosphere was overheated at that time. And it is because of this overheating that you saw this unusual enhancement. So similarly, I used the model for interpreting the transient layer which occurs or which you get to see occasionally in the Martian electron density profile, which we refer to as the M3 layer. I extensively analyzed the MAVEN observation to characterize this M3 layer. That is, what is the typical density of M3, at what time it is occurring, whether it is latitude dependent or whether it has got some dependence on solar radiation and all that. So after characterization, I used my model to understand the reason of occurrence of this M3 layer. So two widely cited reasons for the occurrence of M3 layer is the presence of crustal magnetic field on Mars and also the sudden enhancements in the electron temperature. But using my model, I could see that, I could prove that actually two of these factors might not be contributing much to the formation of the M3 layer, which indicates that some other mechanism is under play for the formation of M3, which we are still in search of. So finally, I used the model for understanding the solar cycle and seasonal effects on the Martian ionosphere, but that particular work is still under review in a journal, so I'm not getting into the details of that particular work. So to summarize, I developed a model, and this model was combined with the NASA MAVEN spacecraft observation to enhance our understanding and interpretations of the features of the Martian ionosphere. So throughout my research work, I had the opportunity to work in collaboration with several international and national institutes which focuses on space and planetary sciences, and thus I was able to contribute towards the globally recognized planetary ionospheric research. So with this, coming to the implication, why we need to study all these factors. Okay, as we all know, India is gearing up, there have been declarations like India is gearing up for next Mars missions. So there is a very high probability that we might witness for another successful mission to Mars from India. So not only India, several other space agencies are planning missions to Mars. So all these results can be used as a useful tool while designing instruments for Martian ionospheric measurements. And moving forward, my plan to study the Venusian ionosphere using the same model, which has got several properties very much in common with Martian ionosphere, and I hope I will be able to come up with interesting results soon. So with that, I conclude my presentation, and thank you so much for patient listening.
Thank you, Dr. Vrinda, and congratulations once again for the award, and wishing you all the very best. May I now call upon our next speaker, Dr. Harish V.S., Junior Scientist and our own KSCST member from the Malabar Botanical Garden and Institute of Plant Sciences, Kozhikode, for his presentation. Over to you, Harish.
Good evening to one and all. I'm Dr. Harish, and today I'm going to present some of my glimpses from my botanical journey. I started my research career from KFRA, then moved to Calicut University for my PhD work. Then I presently working as a junior scientist in KSCST Malabar Botanical Garden, Calicut. For my entire career, botanical expeditions are conducted for all over India, including Western Ghats, Andaman and Nicobar, northeastern states, and some part of Western Himalaya, especially for the families Musaceae, Balsamiaceae, Marandaceae, Zingiberraceae, Rubiaceae, and aquatic plants. These are some of the photographs of the collection trips that I conducted. And I did my PhD work on the anatomical and palynological studies on Indian Musaceae, and that is the first attempt of this kind in Indian Musaceae. And Musaceae is commonly known as the banana family, and it is one of the most primitive family in the order Zingiberales, that is the ginger order. And in the family Musaceae, they have already three genera is there. One is the Musa, then Encita, commonly known as Catuvara. Then other one is Musella, that is endemic to the Alpine region. And these are the number of species reported from India. And from we went to northeastern state, we can see the Musa-like forest. These are the four images. First one is population of Musa chismani, second Sikkimensis, and Musa nagantium and Musa itinerans. That is the population we can see. And there are three types of species, habitats are there. The first one is clump forming, and then that is all medium sized one. Then the second one is short clump forming, and the third one is tall form. You can see that encircled, that is I am standing here, and that is the tallest Musa species reported from India, that is Musa Pushpanjali. These are some of the species that I have collected from my expeditions. And coming to the anatomical studies, it is very difficult to do the anatomical studies of the Musaceae because of it has some resin. So I'm not going to the details. These are the major characters used for the species delimitation, that is the epidermal peelings and also the cross section of the lamina. These are the comparison chart of the species I have done, and some may be two-layer. The epidermal characters are one of the key characters. Some may be two-layer, one-layer, and also up to four-layer species are also seen. And these are the petiole cross-section. One species, you can see that some species have the closed one, and some species are wider angle. And coming to the seed morphology, actually I am focusing on the wild banana. Wild banana having the seeds, and already we have observed four kinds of seeds. The shapes, that is ellipsoid, oblate, ovate, and subglobose. And anatomical works are also done. And you can see that cross section of the seed coat, some outer layer and inner layer, and we can see. And these are the different types of aperture seen in the scanning electron microscope. That is the 6 outer layers and 6 inner type. While we view in the naked eye, we can see the two type of, that is verrucate shape and also this same smooth type. And these are the comparative morphology of the seeds. And these are the surface sculpturing of the SEM analysis. And this is the seeds anatomical work. And coming to the pollen morphology, there is not that much of differences in the pollen compared to other other families of the order Zingiberales. We have only reported 3 patterns, that is the rugulate, verrucate, and the silate. And these are the surface capturing of the pollen grains. And during this study, we change the status of the subspecies level of Musa Musa velutina subspecies Marquana to the species level, and also the Musa cartuana, that is a species reported in the Thailand and in southern part of India. We could collect it from the Andaman Nicobar Islands. That is a new distributed record of this family. And also we identified a new synonym of the Musa bulbiciana variety Andamanica, and this is the species of Musa sabuana. This is named after my supervisor, Professor M. Sabu. We identified different color variation throughout the entire Andaman and Nicobar Islands, and during this period we could establish a ginger collection, Musaceae blossom in Calicut University Botanical Garden. It habits around 32 taxa over 100 accessions. Apart from this PhD, I am also interested in the taxonomy of the gingiveraceae, and also from the gingiveraceae, these are the species I have discovered. Amoma, that is commonly known as large cardamom. These species too are discovered from the Arunachal Pradesh, and the species itself, Zingiber arunachalensis, is also described from the Arunachal, and Globa andersoni, it is considered the possibly extinct taxa, and after 136 years, we could collect it from the Teesta Valley of Darjeeling area. And these are the other species of curcuma, that is a common curcuma. The new species we discovered from Arunachal of, not Arunachal, Nagaland area. Then other Hidikia mechucanum, Zingiber campylanatum, and Zingiber cornigerum, all three are described from the Arunachal Pradesh. Coming to the balsams, it is one of the peculiar and potentially ornamental group of plants, and it is most of the species reported from India and endemic. And from this balsam, so far I have described 15 new taxa, in which 9 from northeastern states and 6 from India. These balsams comes under the family Balsaminaceae. It have 2 genera, Hydrocera, it's a common aquatic plants, and impatiens. And impatiens is the name itself, the species, generic name itself says that the impatient nature of the fruits. You can see once the fruit is mature, we can just touch it, will burst out. That is the impatient, the name impatiens coined by the Linnaeus. And the impatiens, I don't know how many of them are identified, how many orchids and how many balsams are inside in this slide. Actually, the first three are the orchids and 2 are balsam. That's why we can consider as the balsams are the monocot counterparts of orchids. Actually, balsams is dicot and orchid is a monocot. And this is a common balsam we can see in the home garden, Impatiens balsamina. This is the floral parts of the species, and you can see that the variation. These are different species. The petals numbers are different, and coming to the habitat, you can see that this is a high altitude grassland. We can see the terrestrial species, and we can see that actually this one is photo is collected taken from 2012. There's a place near to the Pichi, Olagara. You can see that beautiful land, waterfall, and this is Dr. V.B. Srikumar along with me collected for the collection of balsams here. You can see that some of the balsams in the rocks. And this is from the Arunachal Pradesh. You can see 3 types of balsams are there. And these are some of the other habitats of the balsam. And coming to the Khas Plateau, you can see that the entire plateau is fully covered with the species of impatient larvae. And this some of the photograph from the terrestrial species. And this is high altitude species, impatiens, Hensloviana. You can see that standard nature of the plant, as well as the leaves are more hairy. That's a high altitude species. And you can see that epiphytic species. This one is Impatiens modesta growing on the tree trunks. And coming to the Shola forest, we can see some of the interesting epiphytic species of impatiens. That's Impatiens parasitica. That's the term itself is a parasitic in nature, not in parasitic, but look like a parasite. That is why the term coined by the species name Impatiens parasitica is the nature of this plant. And Impatiens violacea, one of the endemic and endangered species that is only seen in the irregular nature park. And coming to the discoveries, I have discovered 9 species from northeastern India. Not going to the details, Impatiens arunachalensis, Impatiens pseudocitrina, Valongensis, This from the in northern part of northeastern part of Arunachal Pradesh. Neelalohide Haridasani. Then Rohingyans is the term itself described from the Rohing of the Arunachal Pradesh. Sironiana also described from Arunachal Pradesh. Invasion Shiomians is also the newly described, not described, newly formed district, Shiomi district. And Turbinensis is also from the Arunachal Pradesh. Coming to the southern India, you can see that beautiful land, grassland, and this is the Irivikulam grassland. From that grassland, we described invasions Irivikulamensis, then invasions Glabrata and invasions Shashidarani from the Palakkad region, and invasion Modesta. This is the species we can easily seen in the Shola forest of the species of the area. Then invasion Sahyadrika, that is the first species I have described from the Nilagiri of Silent Valley National Park. Then these are some of the other species of balsam. You can, these are the high altitude area. You can see that invasions growing along with some of the mosses and also small grasses with in association with water. That is the typical habitat of the balsam, most of the balsams. Chandrashekhirani also one of the, it is a one and only species of scapigerous species with, you can see the leaves are digitated nature. Then invasion Ambellata also one of the critically endangered species only seen in the Travancore region, Travancore region. Anamudica also endemic to the Anamala regions. Then Platydina, these are the, you can see that the beautiful nature of frilled nature of the invasions maculata. The petals are seen like a frilled nature. Then invasion Tangashi, it is commonly grown in the water stream area. Then again one is invasion Mundroni. It is also one of the endemic species of the Nilagiri Biosphere Reserve. So it is coming to the difficulties in the balsam. It is one of the difficult group to work on regarding the taxonomy. Once we dry, we can't get any floral features from this group, and also the difficult to identify from the herbarium materials after preservation is very difficult. And another thing is one in the superficial view, you can see that the front view, all the species are look like similar, but when we go through the dissection only, we can see the differences. It's other species, invasion Citrina, Pseudocitrina, Lavigata, and Sironiana. The front view only itself we can conclude it is the same or like that, but only after the dissection we can say whether it is same or different. These are some of the snaps of those photographs that I taken during this period. And apart from this Balsamnesia also work on the taxonomic revision of the family, the genus Ophiorhiza. This is one of the medicinally important plant which is used for the treatment of cancer because of it have the anti-cancerous component camptothecin. And these are the nature of the flower inflorescence. This is the morphology. The floral shapes are there. And this species also show the heterostyle. You can see that the arrow indicate the long-styled flowers, and the same species with the short-styled flower. And these are the dissection of the flowers. And coming to the discoveries, and this species of Ophiorhiza codiensis, it is considered as the possibly extinct taxa. After 1930, nobody is collected from the field, but we could collect it in 2019 from the Paidalmala of Kannur district, and this present collection is the collection after 106 years. So that is a rediscovery of this taxa from the outside from this type locality, that is from Kerala. And Ophiorhiza radicans also, that is a species rediscovered after 120 years from the Kolatupura of Chandruni Wildlife Sanctuary. And Ophiorhiza picarensis is also regarded as a possibly extinct taxa, and we could collect it from Mukkurthi National Park of Tamil Nadu after 78 years. And these are the species I have described from Arunachal Pradesh Ophiorhiza debiana. This Deb is the researcher who worked on the taxonomy of the Ophiorhiza earlier. Then Ophiorhiza megalayensis is also described from the Meghalaya of northeastern part. Then Ophiorhiza jacobi, that is a critically endangered species that we described from the Cherudoni area of Idukki district. And Ophioraceae georgii is also described from the Andaman Nicobar, Mount Harriet National Park of Andaman and Nicobar Islands. Then Sahiagrensis is also the species described from Nilgiris of Silent Valley National Park. And this one is Ophioraceae madongensis. Previously, it is reported from only in China, but during our collection in 2019, we could collect it from the Arunachal region. So this should be a new record to the flora of India. It is also published in journal. And along with that, we could collect a variety of the species from Shiomi district, and we publish it as a variety of Ophioraceae madongensis, variety Shiomi. And we also recorded Ophioraceae rugosa variety angustifolia species reported from China, earlier reported from China. We could collect it from the Nilgiris of Silent Valley. That forms the new report to the flora of India as well as Kerala. Then Ophioraceae heterostyla, this species only found in the northern part of India, and we could collect it from Agasimala region. That forms this new distribution record to the flora of Kerala. These are some of the species of Ophioraceae. And apart from this group, I could also describe a species of Pleon, one of the beautifully and economically important, potentially ornamental group of orchid. And this is the species I described. It is one of the interesting species, and it will look like a fungus, but actually it is not a fungus, it is an angiosperm, and its name itself very interesting, Thismia. And because of the color, in Aurantiaca, means in Greek word it is orange. That's why we named it as a Aurantiaca, Thismia aurantiaca. It is only, so far in India, only 2 species of this Thismia is there. One is described from the KFRA scientist from the Idukki district, and the other one is our team described from the Andaman Nicobar Islands. Apart from this, I am also collaborating with the scientists from China as well as the Vietnam, and in collaborating with the Vietnam scientists, we could describe 3 taxa, one Acranthera hongi from central Vietnam, and Billolovia thongi and Billolovia lambdongis also from the central Vietnam. If the China also, some Spiractis clavicola is a new discovery. Then 2 new Ophioraceae, namely Ophioraceae gowlingensis, and okay, these are some of the threats. And we also conserved the species in Malabar Botanical Garden and for establishing ginger collection here. And these are the awards and achievements, and these are the funding agency. I would like to thank all the forest and wildlife department of various states, including Andaman and Nicobar Islands, and V.B. Srikumar and Shashidharan of KFRA, and my previous director in charge and member secretary of Dr. S. Pradeep Kumar, and present director in charge Dr. N. S. Pradeep for the support. And this is my mentor. He gave the full support and encouragement for the study. This photo is taken from the Mechuka village of northeastern states. And thank you for your patient listening. Thank you.
Thank you, Dr. Harish. Again, congratulations and all the very best for the future. We'll wind up the session with a vote of thanks by Dr. Deepu Shivadas, Junior Scientist, Kerala Forest Research Institute.
Respected dignitaries and delegates present here, I hope you had enjoyed to these presentations, one from an atmosphere in another planet and another on a biosphere of our own Earth. On behalf of the Kerala Science Congress Committee, I would like to congratulate Dr. Vrinda and Dr. Harish for their recognition and being the recipients of the prestigious Kerala State Young Scientist Award and Chief Minister's Gold Medal. In fact, it's a proud moment for me because I was one of the earlier recipients of this Young Scientist Award 3 years back. And on behalf of the committee, I thank Dr. Vijay Kumar, Principal Scientist KSYST, for introducing the scheme. It was a detailed introduction. I hope you all had noticed the details of the scheme and you can all apply for this. And welcoming the participants. Thanks to all the delegates who had remained seated here. I could see quite a number of people sitting here even when parallel sessions are going on. And I hope you had all enjoyed the presentations made by the winners of the scheme. And thank you all the delegates who had assembled here, and let's see in the next session. Thank you.
With this, we mark the end of the session. An important announcement for